So in this very short video, I'm just going to introduce you to how to work with fruit flies. Fruit flies are also called Drosophila melanogaster. They're the most common organism used for studying genetics because in genetics we want to study multiple generations, and fruit flies also make lots of offspring so that we can find mathematical patterns. So we're going to work with live fruit flies, and we're ultimately going to look at 3 generations of fruit fly data, and your goal eventually is to figure out what's going on with certain traits, how are they passed from generation to generation. So as it turns out, I've been working behind the scenes to cross a specific set of flies in the original parent generation called the P generation. So I'm just going to tell you which flies that I crossed. But the first flies you're going to actually look at are the babies that they made. So sometimes that's called the F1 generation. F for filium, which is kind of Latin for the sons and daughters. So we're going to look at those flies, and you guys are going to put those flies to sleep so that you can look at their traits and see what they look like. And then you want them to be able to wake up so that some of them can still reproduce amongst themselves and then eventually produce the 3rd generation of flies, or the F2 generation. That's going to be an easier generation to look at because we're going to be able to, they're the last generation, so we're going to be able to kill them kind of in advance, so that makes it much easier for you to look at them and what traits that they have without worrying about them waking up. So, if we're going to look at fruit flies, you're going to want to be able to distinguish between males and females so that you can put some males and females into a culture jar, and so that just in case a trait also is kind of determined by sex, you can also find that pattern. So males, really the key thing you're looking for when you look at the flies is you want to kind of look at their butts, or kind of the back of their abdomen, really. And if they have these dark butts, kind of fully pigmented dark butts, then they're male. These are male flies. And if they either have really light, you don't see that black, thick black pigmentation at all, then it's a female. And if you also see this striping pattern, it's a female. So sometimes students worry that they can't distinguish between this and that. You'll get good at it as soon as you see a few examples, and I'll be happy to help you. Females are also typically a little bigger than males, but you can't really depend on that. You really want to look at this abdomen difference. So let's also just kind of survey some of the different types of mutations that I might show you. I tend to change the mutant flies from year to year, but I'll try to show you a bunch, and then in class I'll try to specify which mutations we're actually looking at. So you can actually order flies with specific mutations, and then you want to cross them with wild type flies. Wild type simply means that those flies kind of look like flies typically do in the wild. So when you cross mutant flies with wild type flies, you can start to figure out how those mutations are passed on. So just some quick example mutations. We are going to see some flies with different mutations to their wings. In some cases, they're going to look like messed up like this. They're really going to like jut out at an angle from the fruit fly. That's what I want you to look for. The wild type variation instead for wings is going to be much longer wings that kind of go back on the fly like this. Okay? Now you also have to be careful when you're looking at the flies, because sometimes when you're using your brush to kind of move them around, you might damage the wings. So make sure that you can see that this is what the messed up wings genetically look like. In some years we even use flies that don't have any wings at all. So the mutant trait might be messed up wings or even missing wings. They really just look like little ants crawling around, versus the wild type trait, which is going to be having normal wings. Okay. Another type of fly that we might see might be variations in their body color. So the wild type trait is to kind of have this brown body color like you see over here. Sometimes the body color might be really black or very dark. And sometimes another mutant trait is to have really, really, really light body color. If you have light body color, sometimes that might seem like it's hard to distinguish, but even the wings look like we kind of took a highlighter and highlighted them yellow, kind of. So you'll really be able to distinguish between that if we have that particular trait. So again, the mutant trait is having a lighter or darker body color versus somewhere in between. And the final variation we might play with is their eye color. So in some cases they might have white eyes or very, very dark color eyes, black eyes essentially, versus the wild type eye color, which is red. Alright guys, so I'm just trying to show you the materials you're going to have in lab to anesthetize the flies. I have some live flies here. You're going to have a little Alka-Seltzer tablet that generates carbon dioxide. And when you drop it into the little bit of water that you see there, you're just going to plug the tube. And then there'll be a little plastic tube that you just quickly put over in between the foam plug and the vial with the flies in it. You'll see that pretty quickly puts them out. But you want to hold it in there for at least a solid minute, or kind of when the Alka-Seltzer stops fizzing, because you really want them to be nice and out. The other thing I've seen with some groups is make sure you only add that little bit of water, just about 2 centimeters up. Otherwise, you might get water in there and then they'll drown. So now that we've knocked them out with carbon dioxide, we're going to keep them knocked out by keeping them cold. So you're going to have cold packs like this, or you're going to have rubbing alcohol bottles that have been in the freezer, so they're very cold as well. And you're going to have a little piece of white paper to put over them. Notice that I'm pressing on the paper to make sure that it makes good contact with the cold pack so that the flies really do stay cold. If the paper is a little bit above the cold pack, then they won't stay cold. So here's your opportunity to sort them, to look at them really carefully, and to identify males and females, maybe separate them into little piles with your brush. You should find with this F1 generation that they all kind of have one particular trait, so it should be pretty easy to figure out what they all look like. So really all you need is about 6 males and about 6 females for each of your 2 culture tubes. So if they start to wake up on you, don't panic. Just get those 6 males and 6 females into the culture tubes, each culture tube, and then you'll be fine. So just to save some time, let's say I've got my males and females, and here I am loading them into my culture tube. So I just brush them kind of with the culture tube on its side like this. Sometimes if they fall directly into the food, then they might drown. So you want to keep the culture tube on its side like this. So if you have flies in your culture tubes, we're just going to kill the rest of the flies in this flask of rubbing alcohol. So you'll just dump them down there and make sure that they all get in the rubbing alcohol. If any flies happen to get away from you and they wake up, that little flask of apple cider vinegar and soap will trap the rest of those flies, so you don't need to worry about them either.